fungi.
Okay, Dr. Siti, we will begin. Yeah?
All right.
Good morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. Welcome to the August edition of the MyMangrove monthly discourse. So we are proud, very, very proud and very honored. Thank you very much, Dr. Siti Aisah. So today we're very honored and glad to have Associate Professor Dr. Siti Aisah Alias, one of the most prominent mycologists, I would say, in Malaysia, to be with us today to share about marine fungi that occurs in our mangrove forest. So this is an aspect of study that is pretty much understudied in Malaysia and perhaps also throughout the tropics. Not much is known, among general scientific community, including as well as for local communities. Yeah? They may know their habitats and ecosystems, but we may not know about other organisms than plants and marine fauna or terrestrial fauna that live within the habitat and ecosystems. A little bit of introduction about Dr. Siti Aisah. Besides being one of the most prominent mycologists, she holds quite a lot of responsibilities within the University Malaya, as well as at national and international stages. So her research interests, basically, she specializes on fungi diversity, physiology, enzymology, bioactivity of marine and polar fungi. Imagine that. She works not just within Malaysia or within the tropics, but extended to polar. Most of the work is in Antarctica, right, Dr. Siti? Yeah, correct. And then she has been a deputy director for the National Antarctic Research Center and Malaysian Antarctic Research Program. Yeah, and that includes soil polar fungi, antimicrobial activity, and cold adaptive enzymes and biochemistry of polar microbes. Yeah, and then under Higher Institute of Center of Excellence, that's the research that she's carrying out under this institute. And currently, Dr. Siti is leading a project on latitudinal differences in response and adaptation of microbes to atmospheric changes. So a lot of work actually from this work, a lot of contributions to our better understanding towards climate change, towards changes happening in our habitats and ecosystems as well. Very important success that I need to highlight now is that Dr. Siti has described more than 20 marine fungi. And then this is, like I told you earlier, this is understudied. So this topic is understudied. So we need experts like Dr. Siti Aisah, and perhaps from this seminar today, we will be able to give birth or to maybe trigger some more interest, Dr. Siti, on this theme or topic of research, particularly to have more experts coming from Malaysia to describe perhaps more species from Malaysia. And I would also like to highlight that Dr. Siti Aisah is a fellow of Academy of Sciences Malaysia, and she is the national representative for Scientific Committee on Antarctic Research in Life Sciences Group. So we have one representative from Malaysia in this very important group. And then Yep, and then through this event today, this is one of the mechanisms in which Dr. Siti participate in disseminating knowledge and through other programs at university or national levels. So there you go. We are very, very proud to have Dr. Siti Aisah today to discuss and to describe to us the marine-derived fungi from the mangroves of Malaysia. So without further ado, Dr. Siti Aisah, you have the screen. I mean, take your time, half an hour, 40 minutes, 45 minutes. Basically, it's your morning, basically. Yeah? The whole morning is yours. So, and then we look forward to discussion at the end of the talk later. And perhaps this will give birth to more collaboration as well in between you and also our colleagues all throughout the universities in Malaysia, and perhaps our colleagues. I see some names. Actually, we have viewers from outside Malaysia as well. So welcome to everyone to this eighth series of MyMangrove Monthly Discourse. So Dr. Siti, you may share your screen now.
Okay. All right. Thank you, Dr. Eldry, for such a lengthy introduction. Okay. And good morning to, I think, maybe some of them are my collaborators, my acquaintances, my friends, my colleagues. Thank you so much for being here together with me, for me to share the diversity of marine mangrove fungi and how diverse they are in Malaysian mangrove ecosystem. Okay. So, don't know why. Okay, correct. So I'm planning to give the talk in this kind of presentation where the first one I'm going to talk about mangroves, and then followed by definition of marine mangrove fungi, their diversity, and also not only in the world, but also, in the world, but also in particularly in mangrove ecosystem. And then it will be followed by early history of marine mangrove fungi in research, and then their habitats, their characteristics, their roles in the mangrove ecosystem, and then followed by factors that contributing to the diversity of these fungi. Then I will highlight some research that have been going on for marine mangrove fungi in Malaysia. And then I would like to discuss or also to project one important topic, which is mangrove as a reservoir for marine fungi with novel secondary metabolites. And then I will talk about the future prospect and also where are the missing fungi. Next. Right, I think since I'm the number 8 for this MyMangrove, number 8 is a nice number, right, Eldry? Okay. So I think by now you're all familiar with mangroves in Malaysia. There are two important points that I would like to highlight here. Okay, mangroves in Malaysia, there are 48 species, which is considered the highest diversity in the Indo-Malayan realm, or probably in the world. I'm not sure about that. Probably the expert can concur with that. And therefore that Malaysia actually ranked as the 6th largest in the world in terms of areas, but for the species, we are very diverse, 48. Yeah. So before I move to many aspects or topics, I would like to define marine fungi based on the history, based on work that scientists working on. I said I'm working on marine fungi. I'm a marine mycologist, and people ask, are there really there's fungi in marine ecosystem? And even until up to yesterday, when I asked my friend to join this talk, they did say that no, there's no fungi in marine ecosystem. So let me bring you to the journey how marine fungi are being defined. Okay, actually, at first, where history is concerned, I started in 1980s, where Sacardo, Alias, and Everhart, they described the fungi just taxonomically. There's no mention about physiologically. Okay, but then followed by publication in 1944, a very famous publication by Bargun and Minder, where it opened the entirely new field of endeavor, and this not only taxonomically but also physiologically. Okay. So where definition is concerned, I would like to follow the 4 definitions through the timeline. Okay, the first one, for the physiological characteristics by Johnson and Sparrow in 1969, they described marine fungi is the species that can grow exclusively in the marine ecosystem. Not only grow, but also to reproduce under the seawater condition. And then secondly, followed by Kohlmeier and Kohlmeier in 1979, who has published a book of marine fungi. This book I've used a lot for my research also when I was a student. They came out with 2 terminology. One is the obligate, and the second one is the facultative. Okay, the obligate is the truly marine fungi, where they grow and they sporulate, and they, when you say about sporulate, meaning they can reproduce exclusively in marine or estuarine habitat. Whereas for the facultative marine fungi are those fungi that actually originally from the freshwater ecosystem, but slowly they found their ways to the marine milieus, and then they are able to grow and sporulate in marine environment. So I give you one of the best example for the facultative marine fungi. That is this Seborrhea verrucosa, which first time being recorded in Japan in the freshwater ecosystem, 1978. And the second encounter, as far as I'm concerned, back then in 1996, we found this Seborrhea verrucosa in Morit Mangrove, Malaysia. So you can see verrucosa, verrucose meaning they have ascospora wall. Okay. Next, then Hyde and Jones, 1988. They define the marine fungi as a group that is based on their ability to occupy specific habitats and substrates. Yeah, I also like this definition because it makes many people Ligniclus. If you can see here, this is the surface of a wood. You can see there's a fruiting body here. Ligniclus referring to fungi that grow not only on the surface of the wood, but they're able to permeate into its fibrous structure. Yeah, and the second one is the Algiculus fungi. These fungi, they can inhabit or associate with marine algae, and this is also very much understudied. Okay, the third one is the Arenaclus. This is the Arenaclus fruiting body. If you can really squeeze your eyes, there's a black spot, few black spot here. This is their fruiting body of Arenaclus fungi. And if you break this round spot, which is quite tough actually, you can discover very fascinating, attractive ascospores with many filamentous and sheaths, which I will show to you later. Okay, the fourth one is the folliculus that fungi grow. On leaves. Yeah, this one. And the last one, last but not the least, this is the group that I'm going to talk more later, is the Mangoliculus fungi, where fungi that grow, okay, exclusively, or that inhabit mangrove forest. But I also would like to stress here, we're talking about marine here. So I'm only going to include all those data, all those findings that fungi that have been described, being discovered, being isolated from mangrove plants, mangrove trees, that being exposed to the seawater during high tide or low tide, and they're able to grow using the sea salt media. Okay. The last one, which I've been using this, definitely I've been using this definition by Pang et Alia in 2016. They said that any fungus that is recovered repeatedly from marine habitats, and of course, for physiological aspect, these fungi are able to grow or sporulate in the marine environments, and also they can have a symbiotic relationship with other marine organisms. And the last one, which never been discussed before, this particular definition stated that it is shown that they can adapt and evolve at the genetic level or be metabolically active in marine environments. Okay. So next. Right. Before we talk about diversity of very sensitive, this computer, I did not touch anything. Okay, before we talk more on their habitats, their occurrence, their characteristic, I would like to bring you to the timeline of diversity of marine fungi. When I talk about diversity of marine fungi, this includes all those 5 categories, ecological group earlier, not only mangrove. Yes. Okay. So in 1979, Kohlmeyer and Kohlmeyer, they listed about 209. And if you look at the journey from 2000 to 2022, there's a drastic increment of number of marine fungi, which is more than 2,000. And this, without a doubt, must be attributed to the advancement of molecular technique. Okay. Next. Okay, there are lots of numbers here, but please bear with me. Let me explain to you. I think something is trying to help me with the PowerPoint, I think. Okay, let's focus on the number here. Okay, today, we have described, or we have reported scientifically about 200,000 fungal taxa, okay, currently scientifically described, which are not many, right? I don't know about how many insects, how many butterfly, how many birds, but I know. Then I also would like, there are 3 estimation number of fungal species have been done by very prominent scientists. The first one is Hawksworth, 1991. He estimated about 1.5 million, but his estimation actually are based on a ratio of plant to number of fungi per plant. And it was based on the temperate region, not the tropical region. And I'm sure by then, if he did the ratio for the tropical plant and fungi, I'm sure he will get even a lot more than this number, 1.5. And then in 2011, Meredith Blackwell, she estimated there are 5.1 million number of fungal taxa based on high throughput sequencing methods. And then in 2017, Hawksworth with his co-worker estimated between 2.2 to 3.8 million. Okay, so where does the diversity of fungi in terms of percentage, if we compare to, we don't want to compare with the estimation, okay? We compare with the, I'm going to compare this with the Blackwell. Okay, so the diversity of fungi, if you follow the number, the percentage, the best that you can gather for 200,000, there's only 8%, or the lowest percentage is only 3%. Okay, from the total number being estimated. And where does the marine fungi stand then? Only 0.0% from the estimated number of fungal species. So there are many more to be discovered, perhaps. So we will talk later about them in the topic, where are the missing fungi. So next, what about number of fungi in Malaysia? Okay, so we follow the timeline. The first earliest list was in 1978 in Graviela Journal, and then followed by 1913 by Brankov. He listed number of fungi from the Federated Malay States with notes on their occurrence. I think the Federated Malay States are referring to the 4 states, if I'm not mistaken, include Pahang, Negeri Sembilan, Perak. Maybe one more is Melaka, I'm not so sure now. Selangor, okay. Thanks, Audrey. So there are about 105 species. And then in 1921, Chip, this is a very famous mycologist. He has collected and described many fungi from Tanah Melayu back then. Okay, he published a list of 861 species from the Strait Settlement. Strait settlements, I think, include Penang, Malacca. Maybe Edry can help me again.
Singapore.
And Singapore, yeah. Okay, and also the Federated and neighboring Malay states. Then in 2012, with my effort together with Dr. Lee Susi from Forest Research Institute of Malaysia, FRIM, Professor Jones, my former supervisor, Nazura, and also Agnes, we came out with a checklist of fungi of Malaysia. It took me about 7 years to come out with this list. There are many reasons and factors for that reason. Okay, we have reported about 3,804 fungi known to occur in Malaysia. So if you compare to the world fungal taxa, it's only about 0.02%. Okay. Next. What about marine fungi from mangrove of Malaysia? Okay. I will talk about the history later. Okay, so the first report was by Jones and Tan, Dr. Tekun Tan from NUS Singapore. The first report actually was from Jones and Tan, not from the mycologists in Malaysia, where marine mangrove fungi is concerned. They reported 32 species. Okay. When I'm being pulled into this project, we came out with a new list. In 1995, we listed about 169 species. Okay, and then in year 2009, together with Professor Jones, we came out with this monograph, Marine Fungi from Mangroves of Malaysia. So there are about 352 species of fungal taxa. So since 2009, I believe there was no checklist from Malaysia, Mangroves of Malaysia, where marine fungi is concerned. However, from 2009 up until today, we have described maybe, I would say less than 10, maybe 5 to 10 new species. So even if there are any increment of marine fungi, probably now we have about 340 marine mangrove fungi in Malaysian mangrove ecosystem, which is a very small portion, which is about 0.002%. Okay. So only 0.002%. Considering that Malaysia harbors 48 mangrove trees, the 6th largest mangrove areas in the world, I'm sure that if we put a concerted effort and work together, we can produce a lot more numbers. But it's not about producing numbers here. Now, I will talk more later about the importance of this research. Okay, I would like to share with you the early history of research on marine Mangoliculus fungi and also my journey into the unknown. Okay, let's focus on Dr. Aisah. I believe many of us know him well. He's a very important mangrove man. Okay, the reason why I'm staging him here because he was the first one to introduce me to mangrove when I was maybe in my 3rd year or 4th year of doing undergraduate studies. Those days, 4 years, yeah, we follow system Fungal. So he took us to the mangrove ecosystem. This is the newest photo, not the photo when he took me to mangrove before, because I believe this is Dr. Gillian Uy at the back. Okay, maybe Gillian not exist back then. Okay, so he took us to mangrove ecosystem in Port Eastern, through the muddy mangrove floor to the stinking smell of the mangrove floor. He stated that microorganisms play a very important role in this very beautiful and healthy mangrove forest. But I couldn't see the microbes. So I was wondering, and I was scared to ask, where's the microbes, Dr. Sasikumar? So I was curious. Okay. And then during my final year, if you can focus on arwah Professor Nawawi, okay, who played a very important role in my career. Okay, so during my final year, I took his course on plant pathogen, and he did say that fungi are friends and foe, kawan atau musuh. So how could that be? And I was even more curious. So that is the reason during my final year, I choose Professor Kutubuddin to be my supervisor. Not him choosing me, I insist that I want to undertake project with him. So I did not work on mangrove fungi, but I work on succession of fungi on Hevea brasiliensis leaves, daun getah. Yeah? Okay. And at the same time also, that time, Professor Jones started to venture into Southeast Asia region. He was a constant visitor to the Department of Botany back then in UM. And between Professor Jones and Prof Kutubuddin, they worked together very well. They published the second, if not the first, report on Malaysian mangrove fungi. This one. So later I got an offer. I think maybe Prof Jones and Prof Kutubuddin trust my ability to further my study. So I was sent to University of Portsmouth in 1993 actually. So the photo here is in 1995, where the first paper that I presented for marine mangrove fungi. So you can see here Professor Kohlmeier, Volkmann Kohlmeier, Professor Guys Jones, my former supervisor, Dr. Kevin Hyde, my collaborator before, and then Dr. Professor Tekuntan here. They're all very, they played a very important role in my research back then. And we are all very close-knit family, like really a family. We know each other, we know their story. So it was, until now, it's a very nice research group. Okay. So now,
and I see Dr. Sundari there as well.
Oh yeah. Dr. Sundari was there for 3 months, if I'm mistaken, and her PhD was on Araneuculus marine fungi. Yeah, fungi that grow on sand. Okay. Next, now getting serious into the science itself. So I will talk about the habitats of this Mangoliculus marine fungi. So please remember again, when I talk about marine fungi, the substrates that we have collected or we will collect, only the substrates that have been exposed or inundated during the high tide or low tide. And they're able to grow in a seawater media. So the habitat, they can be found anywhere, everywhere. The pneumatophore, the prop roots, the fronds, the fruits, the leaves, of course, they're everywhere. They can be found. Yeah. And I would like to highlight also here for mangrove, this habitat, they are so understudied. And I think the last paper to report the occurrence of fungi was in year 2000. And what we have discovered back then, we noticed that the fungi from the Nipe are very host specific. They cannot be found on other mangrove species. So next, right, how they look like in the field. Actually, what you are seeing here, what you are seeing here is not what you get in the field. If you can see, if you can found this in the field, sorry about this, why are they so excited jumping? Okay, this, what you see here, actually is after the incubation, after a few days or 1 week or 2 weeks of incubation. So when you go to the field, okay, all you see is just the twigs or decaying twigs, decaying barks, decaying branches. There's nothing. You cannot see this beautiful fruiting body. So how to promote their growth? So, but then again, you must also able to collect the right substrates. Otherwise, you get nothing, even though you incubate them well, enough humidity and all that. So, the trick is, this is what I've been taught my supervisor, Professor Grace Jones. So the trick is, you take those decayed branches or twigs, using your fingernail, you press a little bit on the surface of that wood. if the surface of the wood is soft, and then also if it's a bit wet, soft and wet, okay? And on top of that also, there could be some dark sign or black sign like this colored, which relate to the sign of fungal activities, because the mycelia activities tend to make the wood surface colored. Some are purple, some are black, some are brown. So with that kind of trick, you bring that to the lab and then you incubate them. When I say incubate is you put in a nice container, you put a layer of wet tissue papers and incubate at room temperature. And with enough humidity, keep your finger crossed, after 1 week or maybe a few days, you will start to discover this nice brown or black dot, this fruiting body growing on the wood. Okay. So then the investigation or inspection of fruiting body begin. Right. But before that, I would like to also mention there are many types of fungi in the mangrove ecosystem, marine fungi. You can find also Basidiomycota, mushroom. But the Basidiomycota in the marine ecosystem, in mangrove ecosystem normally, they're very different than the Basidiomycota, the terrestrial one. Because in the marine, they tend to be very small, much smaller, and the fruiting body also a bit softer. And why their size are so significantly different from the terrestrial mushroom, we think that it could be attributed to the harsh environment with the desiccation, dry, and then water, inundated by water, and then the sun, strong sun, so they have to survive. That's why their fruiting body much smaller. So next, we look at their characteristic, the spores or ascospores or conidia. This is the main reason why actually I was so sure I wanted to study this marine fungi, because when I first being introduced to these ascospores, I thought they were so beautiful. I thought they were so attractive. I thought they were so unique. Those unique criteria that helped me to memorize their long scientific name, because I hate scientific name when I was in my final year, but I survived for this marine fungi. So if you look at this light micrograph, you can see here, this is an appendages. This is appendages. And this is the sheath, okay? The sheath. And this is another appendage, which is very long. After a few minutes, if you leave it, it will unfurl very long. I couldn't measure them because it is very long. So what are the importance of the sheaths and appendages? We believe it is very important for their survival, dispersal, and attachment to the substrates. Okay. Next is the pigmentation. We also observed the marine fungi, doesn't matter, Ascomycota or the imperfect fungi, majority, especially those that can be found exposed to the sunlight, the fruiting body, they are dark, they are pigmented. And this we attribute to the as a protection to the sunlight. Yeah. And because if you compare with this terrestrial imperfect fungi, of course there are pigmented conidia as well, but we can see this is very pertinent in mangrove ecosystem, especially by the coastal region, which is direct exposed to the sun. Okay. Next, now I'm going to talk about the role of marine fungi in mangrove ecosystem. Just ignore it's a simple figure, but what I'm going to stress here, this one particular criteria of fungi that make them deserve to be in their own kingdom is being filamentous. being filamentous in the sense that they have filament or mycelia, we call it, that can penetrate few centimeters into the substrates. And that is why those days between mycologists and bacteriologists, we used to fight with each other. For bacteriologists, they said the bacteria play an important role in marine ecosystem. Whereas the mycologists said, no, it's the fungi because they can penetrate a few centimeters into the substrate and will do their job as a decomposer. Okay, and another thing is those hyphae or mycelia, they will secrete the enzyme. Okay, enzyme, which is very important for decomposition process. So that is why we believe that fungi play a very important role in nutrient recycling and also contribute to the mangrove food chain. Yeah. Okay. Next, now we cannot run from the discussion about what are the main factors that influence frequency of occurrence of mangrove fungi. And they are not only one or two, they are supporting each other. And if we can have these holistic factors, which include the mangrove location, food specificity, water temperature, salinity, and effect of wood porous and all that, with or without bark, I believe that we can obtain higher number of marine fungi and not only about 340 that I have presented earlier. Okay, next. So the following few slides, I'm going to highlight a few research that have been undertaken in Malaysia. And again, I would like to stress this is about marine fungi because I know that while doing some reading, I've also encountered some endophytic fungi work. Professor Moritz, I think he did publish. But what I'm also UPM group also did publish for antimicrobial activities. But here I'm going to talk specifically for marine mangrove fungi. Yeah? So the first one, this is going to be a never-ending research, which is on collection, identification, isolation, and culture of marine fungi, and then followed by estimation and statistical quantification of diversity. And we're not talking about Shannon or the simple indices, but we can talk about PCA and all that to illustrate their community structure. So this kind of research is really need to be done before our mangrove disappeared or before the ecosystem getting more shrink. Okay, next. Right, the second one is involve molecular genetic and phylogenetic analysis. We published some papers. I believe that multi-gene phylogenetic analysis using at least 7 to 8 markers if you want to publish in tier 1 papers are very important. Yeah, and then also using the fungal diversity, using the unculturable, because what I have presented so far, what we have presented so far actually are those culturable fungi, not the one that cannot be cultured. So I believe that if we can adopt the two techniques, the uncultured and culturable, using molecular technique, we can obtain a lot more numbers of diversity of marine fungi in mangrove ecosystem. Yes. And next one is research on antimicrobial potential and secondary metabolites of marine fungi in Malaysia. We did publish some of the finding, and I would like to share here. Okay, I would like to share here. This is the topic that I would like to talk today is on reservoir for marine fungi with novel secondary metabolites. Those metabolites here that you can see actually already being published. But what I'm trying to stress here, these fungi, Hello Rosalina oceanica, Aigilus parvus, Hello Stephania villosa, they can be found in our mangrove ecosystem easily and without fail. Every time I went to the mangrove, I will encounter these 3 species. So there's a statement here. Why marine mangrove fungi, they produce many very interesting compound, and this could be attributed to their habitat, which is considered extreme. And you know, when microbes, when they live in an extreme environment, they tend to secrete very unique secondary metabolites. And these harsh conditions, when we talk about harsh conditions, is the high tide, the low tide, the desiccation, the temperature, and then the UV. This what we are considering what we are talking about is a harsh condition that actually can contribute to the production of many interesting metabolites. Okay. Next, apart from the three that we have encountered in Malaysia, I would like to stress one particular class, the Dotidiomycetes. These Dotidiomycetes is the class of marine fungi. Okay, they are the largest and most diverse class of Ascomycota. And also, according to Hyde 2013 and also Moria 2014, they did mention that the Dotidiomycetes, marine fungi from marine ecosystem, in particularly mangrove ecosystem, okay, through the genome sequence, they have a very extensive array of secondary metabolites. So in my study as well, 40% of the total number of species that we have collected, isolated from mangrove area, belong to this particular class, the Dotidiomycetes. Yeah. So this is an example, Fasciatis poranipe, Diphrolomyces rhizophore, yeah, that we also have recorded. We have found interesting fatty acid. Yeah. So another example is Sacraduela rhizophore. This also part of our finding. And we know that fatty acid can be commercialized for many purposes, like food additives, and then to inhibit the bacterial. They can also be an inhibitor against the skin cancer protein, and they can be feedstock for biodiesel production. So when we talk about Sacraduela rhizophore, 40% of their crude comprised of fatty acid. Okay, so obviously this is only from Sacraduela. There are many more of fungi from this particular class that we should explore. Okay, and next. So now I'm going to talk about the future prospects. I've given you the definition, the occurrence, the factors that involve or determine their number of diversity, how they look like, how to isolate them, and then the research that have been done so far in Malaysia. So now I would like to discuss about the future prospects and also what are the prospects that we should explore as well. Okay, the first one is about the current scenario on antimicrobial resistance. We know that 70% of aureus strains already resistant to penicillin, and the CRE superbug resistant to the most drugs. Okay, and according to WHO, an average of 525,000 children under 5 die each year due to the E. coli strains. Okay, so I think everybody agreed that it is very timely for us to explore what we have in our back garden. This is what I would like to suggest, fungal bioprospecting under this scenario. Okay, and then the next one is, we know that mangrove is under threat. I'm sure over the past various presentation series from January to July, I'm sure many of you have already discussed how much mangrove of Malaysia being under threat. And then also at the same time, they are under threat, but they are not being explored to the fullest where diversity is concerned, in particularly the marine fungi. Okay, so under this topic, I would like to highlight these endophytes. Okay, We all know, I mean, the mycologists know that endophytes are the treasure chest of antibacterial substances. So we should look more for endophytic fungi that grow in marine mangrove ecosystem. So we should do a wider range of mangrove substrates for fungal isolation. So not only roots, leaves, prop roots, pneumatophore, but we should perhaps really focus on the endophytic fungi. Okay, next. Okay, apart from the antibiotic, apart from the mangrove just now, there's another aspect that I think we should also work on. Okay, this is on a strategic source for new and successful commercial product. I did mention that the fungi, marine fungi earlier, actually any fungi, they are very successful as a decomposer because they have those filamentous mycelia, and the mycelia secreted those enzymes, and those enzymes include amylase, protease, lipase, lignase, chitinase, cellulase. And these enzymes can be capitalized not only for bioremediation, but also perhaps for food production, food enhancement. I know many mycologists in India, they are very successful in this enzyme work, where marine fungi, marine mangrove fungi are concerned. Okay, and the last future, not the last, future prospect that I would like also to stress here, because there's not much study being carried out, is on fungal diseases and also phylogenetic studies of mangrove trees in Malaysia. Okay, so these are something that we need to also consider when we study about, when we talk about the future prospect of marine mangrove fungi. Okay, coming on to the next slide. From Hyde et al. 2019, they came out with a future prospects. They have this amazing cycle of 50 ways where we can exploit fungi industrially. So you can easily find this paper online. It's free to download. Okay, the thing is, what we can summarize from this paper is here. The cycle starts with basic biodiversity research. Okay, this is something which is no longer a luxury for Malaysian scientists, isn't it? Especially the academics. Okay, and then from this basic biodiversity research, okay, we go to the culture, place in culture collection. And I think until now, Malaysia has issues, a problem to set up this culture collection. I remember during Professor Kutubuddin, he did submit a paperwork to the ministry, to Malaysian government, to ask for funding to set up a culture collection. But to them, culture is nothing to do with fungi. Culture is about dancing, singing, and all that. Well, this is what Prof. Kutubuddin shared with me back then. Okay, but I think it's very timely, and I know UPM did make an effort to have a workshop or a meeting, and call for Malaysian to discuss about this culture collection. And I hope something good will come out from it. Okay, so this culture collection is the reservoir of genetic resources, which cost is priceless. It's very priceless actually, because this cultures can be used for applied research. And I have already staged or table earlier that current scenario, how much that the antibiotic is no longer working on those pathogenic organisms. So this is actually very timely and very important. So if only we can convince our government for how important the basic biodiversity research is. Okay, next, right. Okay, so I think by now, I hope you understand how important the fungi is. But then you're probably wondering, if it is so important, why we recorded so lesser number, very small number. For instance, for the marine fungi, only 0.02 of the world fungal taxa. Okay, mangrove fungi, marine mangrove fungi. And then where marine fungi, either from mangrove, from algae, from seaweed, from other substrates, only 0.04. So there must be something, I would say something wrong, but there must be something that needs to be done, or a question. If we have estimated there are about 1.5, depending on whose estimation you are using, like say Hawksworth and Lucky, about 3 to 8 million, if I'm mistaken. And then the Meredith Blackwell, she estimated about 5 million. So there must be something that we need to be doing, because we need to answer this question. Where are those undescribed fungi? Where are those missing fungi? So if we look at papers, there are many papers actually, but I would like to highlight these papers by Gareth Jones. He has highlighted some numbers here, some factors, okay, that we should work on. The marine-derived fungi, other group of fungi, wider geographical sampling, non-cultural fungi, cryptic species, whole species specific. But there's one thing missing here, that is the human capital itself that are lacking. And I was actually from University of Portsmouth, where they used to have Department of Botany. Now they no longer have that. Department of Zoology, they don't have that, because they have to cater for the customer, the students. They are into CSI. They are into the applied aspect. So the talent that is missing here. It's a challenge actually for us. Okay, so I think the next is the last slides that I would like to give some conclusion and take home message. Okay, there are only 3. The first one, Malaysia as a country has a greater potential, bigger than what we thought as a mega diversity country. And I was only talking about mangrove ecosystem, okay, which has with seawater influence. Okay, and the second thing is biodiversity finance must be increased, because it pays to invest in biodiversity. And I couldn't see the audience today. Perhaps there are more senior who can fight for us, who can help the juniors. Maybe I'm not junior anymore, but I'm in between probably. Okay, and the last take-home message is about bioprospecting. Bioprospecting actually is a tool to find important potential source of novel biological products for medicine, for food, agricultures, etc., etc. So these are the 3 important take-home message that I would like to share. But before I end my talk, I must acknowledge various industries, various grants, research grants, either from the university or also from the ministry like MOSTI, and also my collaborators, Dr. Rizman, Prof Jones, who have left a footnote in my life, very important, Dr. Hyde, Prof Kalai Pang, Prof Oli, Dr. Kitch, Dr. Teo, and then Luo and the TIO team. And this, I cannot put all the faces here, but I would like to thank them. Okay, so I think Eldri, I think I have ended my talk, and so that is some of the references that I have used. Okay, so can I stop sharing, or?
Sure, you can. We can just stay with the slides.
Or maybe we stay with the slides.
Yeah, maybe you want to refer.
Mm-hmm.
But thank you very much, Dr. Siti Aisah. Ya Allah, this is a very good sharing. In fact, many new knowledge for me myself. Working in the mangrove, I pay less or maybe very minor attention to fungi and to the microorganisms in mangrove, of course, including, not just doing research in pristine area or undisturbed mangrove forest. In fact, there are potential, like you mentioned, towards the end of the human influence, which may actually trigger more activities, yeah, by fungi and microorganisms as well. And of course, you've listed some of the potential substrates or some potential areas within mangrove that should be explored to understand more about the diversity of marine fungi. Yeah? And I like that it is actually a special name, a special category for fungi found in mangrove. So that's very important knowledge for all of us, and perhaps it can be also described, or we can coin some of other special aspect of biodiversity component within mangrove forests. And the basics the basics of the study of fungi in mangrove, or perhaps in other habitats and ecosystems also. And through your explanation earlier, we can see here the priority of fundamental study or basic understanding on fungi biodiversity is declining. So, but then they are the most important foundation, yeah, actually, for us to move or to go on this next levels or steps to explore the potential. So there you go. I mean, I have to ask you, like, okay, if Portsmouth, they don't have faculty of, School of Botany or School of Zoology anymore. But how about you yourself at University of Malaysia? Do you have a good team of successes to your study, or in this team, or in this topic? Dr. Siti, if you can share. Because now I'm worried as well, because it's pretty much the same trend for plant biodiversity and fauna biodiversity as well.
Right. There are two things here that I can respond to it. If you talk about the successor, obviously at the moment I cannot see anybody. Anybody that will take. I have trained few good students, but they all have successfully ventured into corporate sector or in the ministry. I'm happy for them, but like I said, the talent, the successor for this, and marine, especially in marine aspect, because Malaysia is a very important CTI country, yeah, Coral Triangle Initiative country. So we have to have mycologists, if not mycologists, microbiologists, but if we talk about this topic, of course, mycologists that focus on, that will work in marine ecosystem, be it mangrove, sand, algae, seaweed, things like that. Yeah.
Yeah, it is a worry then. So perhaps we will find a platform where maybe we will come up with a strategy maybe soon, because the same issue we face for other aspects or other research themes, yeah, and topics also for mangrove. So perhaps we can start by recruiting or approaching other colleagues as well throughout the country to find or to measure the interest of these particular themes and topics. That, yeah, maybe we have to have a separate discussion on that. But then, okay, so now we open the floor maybe for 10-15 minutes of discussion if we have any question for Dr. Siti. But I myself, I'm very impressed with this new knowledge on the physical characteristic or physical condition in mangrove forest that actually trigger or contribute to them, to fungi, to being more productive. So there you go. Perhaps this is another reason of why they prefer to be in mangrove, and also actually that also contributes to why or how mangrove is acknowledged or understood to be one of the most productive ecosystem. So we always assume that it is just being done by maybe plants itself or themselves, or maybe the animals or microorganisms. So now we know that fungi also actually trigger for the habitat as a whole to be very productive. So I mean, perhaps from your deliberation just now, Dr. Siti, you mentioned about tides coming, influencing them to be more productive, or maybe the salt environment, the salinity actually also contribute to that. Yeah, perhaps. If correct me if I'm wrong.
Yes, definitely. I did mention earlier that if you want to investigate marine fungi, you must make sure the substrates that you are going to collect already being exposed to the seawater, especially during the high tide. I think you can tell the highest level of the and then the salinity as well. Salinity played a very important aspect in this case, because without salt, a truly marine fungi won't be able to grow. And also, if you play around with the percentage also, you will discover that when you quantify the metabolites they produce, and you play around with the temperature as well, the quantification data will be different based on the salinity percentage, as well as the temperature that you are incubating them.
Yeah, that's a very nice experimental design already, because we know, particularly our mangrove, we can see some areas with clear zonation of different plant species in different elevation of mangrove. So I think that could be another angle to look at. And we know the tides reach to separate or different zone of mangrove, and within a month, right? So that could be another angle to look at. And then as well as other areas within these mangrove zones that we can compare the diversity of fungal diversity. Yeah. And perhaps also how about on the leaves and the shoots, like further up, that are not influenced directly by salinity. There's, precipitation maybe, Dr. Siti, could contribute to that as well, I assume.
You mean the seawater mist?
Yes, mist,
Okay.
or perhaps they are not directly exposed or touched by salt water.
Okay, we call those group as aerial part of aerial fungi. So if I'm mistaken, the last paper was, I don't remember, 1980s or 1980s by Kutubudin, aerial part of fungi from mangrove. In terms of list of species, yeah, because I know people like Moritz, people like from UPM, UMS, and I think UMT perhaps also, they did study, but they are working on the aerial part of the mangrove.
Yeah, that's right.
Dr. Audrey?
Yeah, why? Oh yes, Maisah, I see.
Yeah. Hi, Dr. Siti.
Hi.
Nice to meet you. I was very excited to learn, from zero to hero today about fungi. Thank you so much for your presentation today. And last time, I also we chat about this fungi as well, because at UMT we had in our team, because I'm studying the mangrove ecology basically. So there's one student basically I do some research about pathogenic fungi, which we also discovered several species. Interesting story about fungi, which these species are basically the common pathogenic fungi for the commercial plants, the terrestrial plants, which we found at the symptomatic foliar or mangrove leaves in situ and also curtain mangrove. So it's very interesting because it is very small study we just started, but I think there's a huge opportunity to explore in future regarding these fungi studies. Of course, Dr. Siti mentioned about the positive or ecological roles or function of fungi in mangrove, but at the same time, we cannot deny that there are some group of fungi that becomes pathogenic to, of course, to the commercial plants that people have studied.
Nature education and community-based conservation.
Carry on.
Sorry?
Carry on, Dr. Aisah.
Yeah. Okay, so basically, maybe because Dr. Siti has mentioned something about the capacity of freshwater or terrestrial fungi to survive in such very extreme condition in mangroves environment, because we know that the main protector for mangrove itself, I think the salinity, that makes the environment pretty much difficult for the freshwater species to supposed to survive. But still, we can seize and found some pathogenic species that is much related to the terrestrial environment. So how do you see this, Dr. Siti?
Okay, actually we did publish some papers on that aspect. If you can try to Google Phylogenetic species. This fungi also can be found in mangrove, on mangrove leaves or in mangrove leaves. And also there's another group of fungi, insect fungi. Did you ever encounter? Because I found insect fungi on the leaves of mangrove, where they leave those brown spots. They won't kill the mangrove trees, but perhaps they reduce the ability to perform photosynthesis, because the brown spot can no longer perform that role. So I think where mangrove ecosystem is concerned, a very pertinent pathogen are those that cause dieback. Okay, mainly from mangrove of Avicennia. But if you want to venture in particular for plant pathogen, like I said, you try to look for our paper on Phylogenetic, and also try to focus also on that insect fungi, because like I said, they are not commensalism organism. They are not really pathogen because they don't kill the trees, but to a certain extent I still consider them as a plant pathogen, because they slowing down the photosynthesis process, even though there's no data, there's no experiment on that.
Yeah, very good point, Dr. Siti and Dr. Maisah, for raising this point, because when I did my canopy gap study before, we were arguing about causes of these nature disturbances on how mangrove create canopy gaps. So one of them, actually, we came up with this theory or maybe suggestions about pathogenic effect caused by either insects or fungi, or maybe other microorganisms. So we are, I mean, the theory is still there. No one is taking the idea of actually exploring this or understanding this properly. And so yes, it is actually a very valid theory or hypothesis that can be explored. And I like that point also. Just before Dr. Maisah entered with this, raised this point, there are also, I mean, we know there's the 40 to 50 true mangrove species occurring in mangrove in Malaysia, but relating to the previous point about the ability of some fungi to also can be found also on the aerial parts of mangrove. This actually relates also to other types of aerial plants like mistletoes, like ferns maybe, or other parasites or saprophytes living on mangroves, right? So there must be some connection with the habitat as well. They may not prefer the saline environment, but they would prefer the special features or special characteristics of other plants living in the aerial parts of mangrove. So yeah, a lot of things to explore. Dr. Maisah, do you have more people interested on this topic at UMT? Do you want to?
Yeah, I do have in the team Dr. Dahlia Wati. She's basically the plant pathogen what they call it, plant pathogen the people who study
Pathologist.
pathologist, yes. So yeah, so she is basically plant pathologist that's studying the plant disease in commercial plants. But when I brought her to Mangrove inside campus and she saw this is pretty much similar to what she sees, somewhere else with the commercial crops. I think that's how it started. So she's still with us. But it's very difficult, as Dr. Siti said, the funding and everything. And then we had one student, graduated with this project. She's, and then, but to encourage her to continue with PhD and so on with very limited budget, I think that is the main anthropogenic, threats to our study lah. I mean, in Mangrove itself, but we try to survive, and then probably we will try to contact Dr. Siti for future collaboration, inshallah.
Yes, you are most welcome.
You must do that, Dr. Aisah. So let's begin.
Audrey, I think there are some.
Yeah, that's right. I just saw a couple of comments on the chat window.
But I think Dr. Harinder raised his hand earlier.
Oh yeah, he did. Okay.
Yes.
Okay, we will go to Dr. Harinder. Dr. Harinder, are you still around?
Yeah, I'm here.
Okay, all right. So let me just read these a few comments first. Hang in there. So Osman Bojo, very good and informative presentation. Congratulations, Dr. Siti. Less than 4% species are scientifically described. And in his opinion, the biggest threat to our biodiversity is not only the anthropogenic activities, which I want to, touch a little bit later on, but also the lacking of taxonomies. Yeah, I think we all agree earlier. Yeah, taxonomies are now considered as endangered species of the world. So not just for fungi study, but also on plants, fauna, like we discussed earlier. Ken Taeyong apologized. He had to leave earlier, but then he is very thankful to the lecture. One question from Dr. Rozaimi. Dr. Rozaimi is asking, do we know how much marine fungi had contributed to the decomposition of detritus and nutrient cycling in Malaysian systems? Do you want to respond to that, Dr. Siti? You did touch on it just earlier.
Yes, right? Yeah. Thank you for the question, Dr. Rozaimi. Okay, I cannot stress it enough how important or how much each individual of fungi play this decomposition process in mangrove ecosystem. When I said there are about 340 marine fungi have been described today, sehingga hari ini. Okay, so I believe all those 340 did contribute or did play a very important role as a decomposer. If we talk about leaves rich in cellulose, the group of fungi that normally really actively decomposing those substrates, leaf substrates, are those chytrids. I did not give an example, but the chytrids. And also the imperfect fungi. I know that the term is very technical, but just focus on there are fungi that actually play very important roles as decomposer on the leaves because they like cellulosic material. So leaves and twigs rich in cellulose. But if you talk about the branches, those fungi that can inhabit the woody material, the ligniculus marine fungi. So all of those individual, all of those 340 species of fungi, they play a very important. But then again, depends on what substrates, because depends on what enzyme they have. Yeah. So I hope, Rozaimi, I am answering your question.
Yeah, I think it's clear to me.
Okay.
Thank you very much, Dr. Siti, for responding. Maybe we can call Dr. Harinder.
Yes, Dr. Siti, thank you for the talk. It's very interesting. I remember I used to take Professor Gareth Jones to the Kuala Selangor mangroves at one time to collect his, twig samples. And my question is there any way of quantifying the density or biomass of these the marine fungi, let's say in mangrove soil? Because the reason I'm asking is we can quantify the stages of disturbances, within mangroves by looking at indexes of plants and animals. But I'm just wondering whether we can add another factor where we can use the density and the biomass. Of course, you can use the species, but I'm just looking whether you can. Is there a method of quantifying the density and biomass of this fungus to determine disturbance levels?
Okay, Dr. Harinda, thank you for your question. You are talking about in situ, right? To measure the biomass is, to my limited knowledge, I think is a very tough thing to do. But if you're referring to the lab experiment, whereby my group also have isolated soil fungi from mangrove sediment. So we isolate, we use walk-up soil technique to isolate those soil fungi. And we can only quantify their numbers of occurrence. But if you talk about biomass in situ, in the mangrove ecosystem, I think it's very hard, very tough, or almost impossible. Did I understand your question correctly?
Yeah.
Yes.
I think you to find representative for one particular forest spot. So maybe we need like more sample from throughout one forest area. Why I think I can relate to what
Yeah.
Narinda's question.
Yeah.
Like for example, like us measuring the amount of carbon in soil, for example. So taking samples from 3 or 4 or 5 different spot within the same forest patch, we will get different number or different numbers altogether, right, from all 5 samples. So I would imagine, based on this knowledge today, they are dispersed all throughout the forest, and maybe some of them are very close and site specific. So that makes it very hard, I reckon, Dr. Harinda, for us to generalize like the biomass for the whole area, unless we solid or super massive samples. To do that, I'm pretty sure we need more people, at least 20 individuals for one particular forest spot. Siti.
Dr. Aldri, if I can add some more. Dr. Harinda, one thing about fungi, they only produce biomass when the condition is suitable. Otherwise, they will be in a dormant state, dormant situation, either in a spore condition, or even case of their mycelia or hyphae, they can just like frozen, hibernate until there's something to provoke their growth, then they produce.
That's interesting, because perhaps the units of measure may be different for fungi as compared to plants and animals. Your units may be different, whether there's a concentration of spores per unit area, or per concentration of your mycelia per unit area. That could be a measure of your density. And perhaps the biomass may be a little bit different because they're too light.
Yeah, I think so far up until today, we measure the occurrence, number of occurrence, the frequency of occurrence. But then those things, the results can only be obtained once you incubate them in the lab. It's not in the field itself.
Alec is responding to the discussion. Alec Liu is responding to this discussion now. So he asks, how can we revive marine fungi if they're dormant?
Okay, if you talk about tropical fungi, tropical fungi love humidity, they love temperature. So what you should be doing is to provide them those parameter. Yeah.
So boost them with our humidity and temperature.
Yeah, and of course,
how about nutrients, the Siti, or we just like provide them with some suitable substrate. And then
okay, if you talk about pure culture, if about ligniculus fungi, mangrove fungi, they like corn meal agar with seawater. But if you talk about soil fungi in mangrove sediment where there's an impact of seawater, they like potato dextrose agar with seawater.
Yeah, that's interesting. Yeah, I promise Dr. Siti, Aisah, 1 hour and 15 minutes talk. But please give me this opportunity or bonus lah, Dr. Siti, for one last question. So human development, human influence on the habitats and ecosystems. How pollutants entering our water system now through maybe from factories upstream in the river, through the rivers, maybe effluents coming from in industrial area, agricultural area, aquaculture nearby mangrove. Will that have some kind of influence to the habitats? And how about us, like working in a mangrove, trampling the soil, taking samples, or maybe also activities like forest restoration? Will that have impact? And how these all of these, like pollutions, and human encroachment, or even just us entering the system, will that have effect on them, on fungi biodiversity?
Right. We talk about the scale. Are you talking about large scale or small scale? Even when I
Small to
go
large.
to the Antarctic or Arctic, the impact of just my footstep may give a negative impact on the population of the microbes there. So where mangrove is concerned, tropical region is concerned, where the soil sediment is concerned, we know that within a small teaspoon can harbor a few hundreds of microbes. But we talk about fungi here, yeah? And to me, where mangrove is concerned, if you talk about larger scale of human activities such as encroachment of the mangroves, definitely will give impact to the diversity of marine fungi. But then again, if you just like me going to the mangrove, admiring it, collecting a limited number of samples, did not pull the whole trees, the whole plants, I think we are doing fine.
Excellent. Thank you
Because
very
this
much.
is tropical, yeah.
Exactly. Thank you very much, Siti. I mean, this leads to the point of us, maintaining, trying to protect or preserve our natural habitats and ecosystems. That gives more. Yeah, of course, we need more exploration on what we have in our mangroves, particularly for the taking this theme today on marine fungi within mangrove ecosystem. But the point that you clearly stated right from the start, that they are host specific, they are very site specific. So the more, I also always insisted, the more areas, the more pristine the habitat, we will be able to keep more species, to store and to allow more species of plants, animals. Now we know fungi diversity as well. And that's just, what we know here in the tropics, in Malaysia's mangrove, only 400 plus species. I'm pretty sure there are more, right? If we have more, and 0.02% is a challenge actually for all of us to fulfill that theory or estimation of 5.1 million, yeah, Dr. Siti. So there you go. So estimates, 0.02% is the knowledge that we know for mangrove. So there you go. That's a challenge for all of us. To end, perhaps all of us that are viewing this discussion today, let's spread this knowledge. Let's for scientists in particular, for my academic friends, let's recruit more people to study this topic, to know better of this area. And then of course this will contribute, yeah, to encountering or to exploring and to come up with knowledge for the users, and we know, their ability to for medicinal purposes in particular, yeah, and as well as to fight diseases, emerging diseases, emerging perhaps pandemic in the future. We never know, yeah. So again, thank you very much, Dr. Siti, Aisah. This is very precious. I cannot thank you more. For your time especially, yeah, and for your to get Dr. Siti is super hard actually, ladies and gentlemen. So this is very precious moment for all of us. And again, thank you also. I can say, Dr. Sasi's, Prof Chong's generation were one of the first groups or generation to explore this habitat ecosystem. And I reckon you were there with Dr. Harinder, right? Supporting this generation, and hopefully many of us today will continue this legacy of understanding mangrove better from all of our favorite themes or topic, and perhaps more new generation of researchers will join us soon. Yeah, and Dr. Siti will be in touch. Yeah, for more in the future. We have already discussed actually through our phone conversation of some ideas of publishing, of producing, and to disseminate, to find other creative ways to disseminate knowledge and information. Yeah. Okay. So again, I would like also to thank everyone participating today, through Zoom and as well as on YouTube. I cannot reach to YouTube at the moment to get if there's any question, but I hope we can deliver it to Dr. Siti. If you have, you can always email her. And then okay, all right. So again, all right. Thank you very much, Dr. Siti. You can unshare.
Okay, stop sharing this.
Yeah, you can stop share now. So and then let me just go to the next in there. Oops. Okay. Oh, am I sharing my screen? No. So all right. So next. Oops. Yes, next we will have Dr. Rozaimi. Yeah, we saw him through the chat just now. So next month, yeah, in September edition of MyMangrove Monthly Discourse, we'll wait for the call, wait for the announcement. So we will be with Dr. Rozaimi, and Dr. Siti Aisah earlier mentioned about microbial activities in mangrove also briefly. That will be discussed perhaps by Dr. Hazlin in November. So more interesting topics coming up in our series. So stay tuned. And again, on behalf of the team of the whole MyMangrove family, we would like to wish everyone a happy Merdeka Day, especially for all Malaysians viewing and being with us today. Selamat Hari Merdeka, and to everyone, happy mangroving. Thank you very much.
Selamat Hari Merdeka.
Thank
Stay
you,
safe, everybody.
Yep.
Thank you.
Bye-bye.